All right. Well, as promised, we're going to be dealing with the cell cycle again. And I just wanted to point you back to this diagram that I drew for us on Monday. And so in this one, it's mentioning mitosis as that final step, or the final portion of the cell cycle. One thing I didn't point out on Monday that I'll mention now is that mitosis and cytokinesis are actually two separate processes. So mitosis and meiosis as well are division of the nucleus. Cytokinesis is division of the cell. And so those are technically two separate processes. Cytokinesis happens at the same time as the last portion of mitosis and meiosis, usually, but they don't actually have to happen together. And then the rest of the cell cycle is referred to as interphase. So everything other than mitosis or meiosis is referred to as interphase. Now, on Monday, we talked a little bit about the processes that happen during these steps, and I talked about the checkpoint. So we have a checkpoint right here at the end of G1 to ensure that the DNA is not damaged beyond the point of being able to replicate it. And then there's another checkpoint here at the end of S. And if there are any copying errors that are made that can be fixed, they will be fixed during G2 to ensure that both chromosomes are repaired. But if at that checkpoint here between S and G2, the cell determines that the DNA replication was so wrong that it can't be fixed, then the cell won't progress past that checkpoint and will go through apoptosis, programmed cell death. And then so over here during mitosis or meiosis, you have another checkpoint called the spindle checkpoint. And during that portion, the cell is going to check to ensure that the homologous chromosomes are attached correctly during meiosis. During mitosis, there's still that spindle checkpoint, but it's just ensuring that the sister chromatids of each chromosome are both attached to a microtubule and aligned correctly. And so as we begin to put the process of what's going on with the chromosomes into the context of the cell cycle, I just want you to continue to think about this cell cycle. So what we talked about on Monday and what we'll actually start our time on Wednesday, continuing to talk through this DNA replication, is S phase. And so in human cells, each of the 46 chromosomes is duplicated, and so then you generate two sister chromatids that are attached together, copies of each. So each homologous chromosome, for let's say chromosome 1, your maternal and your paternal chromosome, each of those chromosomes would now be made up of two sister chromatids. And so at this point, during the entire G2 phase, there is twice as much genetic material as there ought to be. And that's why G2 is basically just a preparation phase for meiosis or for mitosis, where the cell checks on any or fixes any errors in the copying if the cell actually can fix those errors. So let's look closer at this checkpoint during meiosis. And again, there's still a checkpoint during mitosis. It's a little bit different, but this is called the spindle checkpoint. And it's another way of regulating the cell cycle, but now it's a process of regulating meiosis. So if the only cells that actually go through meiosis in the human body are germ cells. And I mentioned this when we talked about meiosis previously, but germ cells can go through mitosis or meiosis. If they go through mitosis, they generate more germ cells. If they go through meiosis, they generate gametes. Male gametes if they go through spermatogenesis, female gametes if they go through oogenesis, but this process of meiosis produces gametes that are haploid compared to the diploid germ cells. And so in this spindle checkpoint in meiosis, the cell is checking for this process of recombination. It's checking to ensure that these homologous chromosomes are attached together, that recombination that takes place at the end of prophase 1 and continues into the beginning of metaphase 1, it's ensuring that those homologous chromosomes are actually physically attached together. And so if that recombination did not happen, that is, those homologous chromosomes are not physically attached together, this will, you can see this here, negatively regulating. It will actually shut meiosis down. It will not allow that process to finish. And so during meiosis, the cell, again, it's checking to ensure that those homologous chromosomes, the paternal and the maternal, are physically attached together. And so you could look at it another way, as if you're checking to make sure that recombination has happened, and if it doesn't, this is going to negatively regulate the meiosis, it's going to shut it down. Or you could look at it as if that reciprocal meiotic recombination has happened, that's been activated, this is going to positively regulate meiosis. It's going to allow it to continue. And so this is the cell's way of trying to protect itself from misalignment. And a couple of weeks ago, when we were talking about meiosis and talking about Mendelian inheritance, we talked about that, a number of these conditions where you have more or less sex chromosomes don't generate large problems if you can treat them with hormones, other than oftentimes the individual is sterile. And that's because at this point, when it goes through the spindle checkpoint, there's no homologous chromosome to line up. So if you have 3 X chromosomes instead of 2, 2 can line up side by side, but the third one doesn't have a pair. And so again, the cell's able to recognize that chromosome is it's not recombined with a homologous chromosome, and so it shuts the cycle down and the cell goes through apoptosis. Or if you had 1 X chromosome, and so you had Turner syndrome, you'd have you'd have an X chromosome without a pair, without a mate to actually have recombination happen. And again, it would fail this checkpoint and the cell would enter apoptosis. And so there is a question as far as, well, how does a cell that fails this checkpoint actually still progress through into where you wouldn't actually have a sterile individual? And so in that case, it's probably a faulty, the machinery that does this check is probably faulty. And so that there's basically no way for the cell to even do this checkpoint. So the cell always progresses through this checkpoint and finishes the process of meiosis. And so let's talk a little bit more about what's going on with the chromosomes during this process. So again, recombination, when the homologous chromosomes actually attach, forming a holiday structure, holiday junction here. You can see that here where you form a tetrad. You form, or a bivalent structure where you have 2 separate DNA or 2 separate chromosomes attached together. And so this process, again, it starts in prophase 1, continues through. There's a stage not mentioned here, and that's prometaphase, when the chromosomes are moving. They've attached to these microtubules here, and they are moving towards the midline, the equator of the cell, but they're not there yet. Once they're on that midpoint, that equator, now we're in metaphase 1. And at the beginning of metaphase 1, these recombination events, these holiday junctions are fixed by splicing these either here or splicing them there. And so again, the checkpoint is checking to ensure that this is actually happening. And the way it does that, if you look here, there's a set of microtubules attached to one of these chromosomes made up with sister chromatids. There are microtubules attached to the other set of chromatids that make up this chromosome. And so there's not, in mitosis, you would have a set of microtubules attached to either side. So this side at the centromere would have a set of kinetochore microtubules, and then on the other side as well, you would have a set of kinetochore microtubules. But during meiosis, you actually only have one set of microtubules attached to each chromosome rather than two. And so the way it would ensure that recombination is happening is that these chromosomes remain aligned when these microtubules change length. When one of them lengthens, one of them shortens, to pull this this homologous pair over to the midline. And so if they don't remain attached together when those microtubules are pulling them and moving them towards the midline, the cell knows that recombination has not happened, and so it fails that checkpoint, and the cell does not progress past there and enters into apoptosis. And so at the beginning of metaphase 1, the machinery that splices these strands to eliminate this holiday junction splice this. And so once that happens at the beginning of metaphase 1, we are actually ready to go into anaphase 1. And so I want to talk a little bit more about these kinetochore microtubules. You can see this reference to the kinetochore microtubules. And so microtubules are a cytoskeletal element. They function in a number of ways inside of the cell. They help maintain cell shape. They anchor the organelles in place. They provide roadways for motor proteins to move on, to move vesicles throughout the cell. And they also function in cell division. And so all microtubules grow out of what are called microtubule organization centers. And so we refer to those as just MTOCs, which stands for microtubule organization center. And there are a number of different microtubule organization centers. One of those is the centrosome. And so if you'll remember back to cell biology, we talked about the centrosome and centrioles as something that actually duplicates prior to cell division. And so the centrosomes are typically duplicated either during S phase or they are duplicated during G1 in preparation for duplicating the genome and entering into cell division. So these centrosomes are a microtubule organization center. And so these kinetochore microtubules here actually grow out of these centrosomes. There's one at either end of the cell, either pole of the cell. And so you grow a number of microtubules out of those. Some of those microtubules are just going to function to stretch the cell so that when the chromosomes start to move to opposite ends, the cell is stretched out and it's easy for the cell to finish going through this process of cytokinesis, of cell division. Some of them form the kinetochore microtubules here that attach to these chromosomes, to these homologous chromosome pairs, moving them to the equator and then ripping them apart during anaphase 1. And then some of them just support the cell and keep it from collapsing on itself as the cell widens and stretches out. But these kinetochore microtubules, these are probably the most interesting and probably the most important in this process of cell division, because they are those that are going to execute the checkpoint to ensure. Hang on. Sorry about that abrupt pause. One of my children needed something. And so, yeah, again, these kinetochore microtubules are ensuring that the chromosomes line up during metaphase, and that the chromosomes are separated during anaphase. And so a couple of more terms I want to point out to you. Bivalent. Bivalent are chromosomes that are in pairs. And again, this is something that the checkpoint is checking for. And then univalent are chromosomes that are now separated on their own, although you still have two sister chromatids at this point in anaphase 1, which will then be separated from each other during meiosis 2. And so one thing I want to remind you of, if you don't remember this from cell biology, is that when your chromosomes condense in preparation, or actually during mitosis and meiosis, when you can actually begin to see these chromosomes, what that involves is taking your DNA and wrapping it around these nucleosomes here. So the nucleosome is made up of a complex of proteins. It's an octamer, a protein that has 8 separate polypeptides to functionally make up these beads, and then has DNA wound around it about 2.5 turns around each bead. And so this is one of the ways in which you can condense the chromosome down to get it to a point where you can't express genes inside there. The machinery that would work in gene expression, which we'll talk about next week, can't actually get access to this. You also can't access this with DNA polymerase to open it up and to begin making copies of this either. So this is a nucleosome, and the nucleosome contains this protein octamer as well as about 2.5 turns of DNA. Here you can see the relative size, about 10 nanometers. And this is just one way of condensing the DNA. The DNA is then further condensed with additional materials. But I wanted to point this out because a portion on the chromosome known as the centromere, which I mentioned a couple slides ago, whereas the point at which the kinetochore microtubules attach, at the centromere in the chromosome, one of these proteins, H3, which you can see makes up a portion of both parts of this octamer bead. This H3 in the centromere, that region of the chromosome, is a different version of that protein. And that's what the kinetochore microtubule is able to recognize as the point at which it should attach. And so whether you're talking about mitosis or meiosis, those kinetochore microtubules attach at the centromere. So centromere is the middle region of the chromosome where the kinetochore microtubules attach. The centrosome is a microtubule organization center where the kinetochore microtubules grow out of. And I know the two terms are very similar, and it does cause some confusion, but I didn't make the terms up, I just use them. And so here is the centromere again, this central region of the chromosome, and here you can see these H3 is referring to that protein in the histone. And again, in the centromere region, it is a special form of H3 that these proteins recognize, and these proteins make up what's called the kinetochore. And so if you look into here, this inner portion here, this purple portion here is the centromere, where you have this different form of H3. And then you have this machine that forms on top of the centromere called the kinetochore. And then you have the kinetochore microtubules that attach to it. And just look, each one of these is a separate protein. So here's one complex made up of 4 polypeptides. Here's another complex made up of a single polypeptide. Here's another protein complex made up of 4 polypeptides, 2 polypeptides, 1, and 4 more, just to form this inner kinetochore, this machine that attaches onto the centromere. Here's your outer kinetochore, more protein complexes made up of multiple polypeptides, forming the outer kinetochore that is physically attached to the kinetochore microtubules. And so you're talking about a couple of dozen at least different proteins responsible for ensuring that these chromosomes attach to these microtubules, to these kinetochore microtubules, all to ensure that the cell can check and make sure that the chromosomes, the homologous pair, the homologous chromosomes are attached together, and to ensure that the chromosomes can be separated from each other during anaphase 1, and then the chromatids separated from each other during anaphase 2. And I mentioned this a couple of slides ago that during mitosis, you have kinetochore microtubules physically attached to both sides of your chromosome, to both sister chromatids. You don't have that in meiosis 1. Remember that you have one set of kinetochore attached to each of the homologous chromosomes. But during meiosis 2, you do get the same format that you have in mitosis. And that's why meiosis 2 is very similar to mitosis, except you only have half the amount of DNA to begin with. And instead of having 46 chromosomes, you only have 23 chromosomes that the sister chromatids are being separated. So here's the kinetochore, here's the centromere, here are collectively the machines responsible for ensuring that these microtubules are attached correctly. And the only thing we still need to talk about in context of what's going on during meiosis at the chromosomal level is how do these microtubules actually move and pull the cell around. And so there are actually two pieces going on. This is out of a genetics textbook, and it's showing you some research that was done to try to figure out where are the microtubules, the kinetochore microtubules, actually shortening. And so you can see here they actually stained a portion of the microtubules in this process and then watched it during the process of anaphase 1 to see if this stained region moves towards the centrosome or actually stays in place, and this portion of them shorten. And that's what they actually found, is that the stained portion, this marked region, stays in place, stays in one location, and this portion of it actually shortens. So where the chromosomes are shortening is where it's physically attached to the chromosome, which is really interesting that you have this microtubule that's grown by polymerizing these tubulin subunits. It's grown to a point in which it can physically attach to the chromosome at the kinetochore, and the point at which it grows, that's where it's actively shortened. So that's one part, one component of it. These microtubules are shortened as the chromosomes are pulled to opposite poles of the cell. Another portion of it is, if you look right here, there's a motor protein. And so if you remember back to microtubules functioning as roadways in the cell for vesicles to move on, you have motor proteins called kinesin that grab onto the vesicles and then walk along the microtubules, moving vesicles around. Here you have a motor protein similar to that, where it's physically attached to the kinetochore and it walks along the microtubules. But in addition to it walking along the microtubule, this microtubule is actively being shortened. And so you have two things involved with moving these chromosomes around. The motor protein actually functions to move the chromosome this way, away from the pole. And that will happen if, as it is attached together, sometimes it will need to push it to get the chromosome at the equator during metaphase 1. And then the process of actually shortening this and pulling it towards the pole involve removing these tubulin subunits that make up the microtubule at this point of attachment. And so once that starts happening, this motor protein will detach, you'll remove some tubulin subunits, and then it'll reattach. and then it'll detach, and you'll remove some tubulin subunits, and it'll reattach. And you'll just keep doing that process over and over and over again until these chromosomes have been pulled all the way to the end of the cell, and anaphase ends, and then telophase begins of rebuilding the nucleus. And then usually going along at the same time as telophase is cytokinesis, where then the cell divides into two. And so this whole process is very highly controlled. The cells have a checkpoint to ensure that the homologous chromosomes are attached together, that recombination has happened. There are complex machines that ensure that the kinetochore microtubules attach to the chromosomes at the correct place, so that when they pull, and those microtubules start to shorten, that the chromosomes are pulled correctly to the opposite ends of the cell. And so with non-disjunction, we talked about that when we were talking about inheritance. In non-disjunction, either the hold at one of the kinetochores fails, or the recombination doesn't end in time, and so both homologous chromosomes get pulled to one end of the cell. And so then what you end up generating is one daughter cell with no chromosomes and one daughter cell with two chromosomes. Either way, you have abnormal chromosomal number, which is known as aneuploidy. And I'll go ahead and write that out for you so you can see what that looks like. Aneuploidy, I think it's EU, might be UE, but I'm pretty sure it's EU. Aneuploidy, and this is an abnormal chromosomal number. And so you can have polyploidy, which is too many chromosomes, or you can have too few chromosomes, but either way you have an abnormal chromosomal number as a result of that non-disjunction. But this whole process is very highly controlled to try to eliminate that from happening, so that the germ cells produce daughter cells, produce gametes that are correct, that are haploid, and that are ready to be involved in the process of fertilization. So I know this lecture is a little bit shorter than the ones that you've been watching, but we're going to need to spend some time on Wednesday continuing and finishing our discussion on DNA replication, and then we'll talk through this, what's going on at the chromosomal level during meiosis, and we'll be in a good position to discuss a paper on Friday, and then to next week begin talking about gene expression. So hope you enjoyed this talk, and even if not, I hope you learned something. Let's go ahead and keep those.